Can I think about it? Because I'm thinking. No, it's public, then we don't
No,
get
maybe
it.
if you want after the streaming, we can
Is it possible to start? Is it possible?
Yes, we'll turn off the light. Ahí estoy, ¿me ven? Ahí. Bueno, disculpen la demora. Bienvenidas, bienvenidos, bienvenidas y bienvenidos también los que están por YouTube. Tuvimos algunos temitas que, bueno, como siempre, hay desafíos para cada año. Bueno, este es el primer de 2023, tenemos algunos cambios, estamos apostando por la presencialidad y la discusión acá, in situ, así que los invitamos a todos a venir y a aprovechar que podemos estar acá. No va a haber más participación activa por Zoom, sí por YouTube. Por YouTube recuerden que tienen la posibilidad de hacer preguntas por el chat, que las vamos a leer a los presentadores y van a poder contestar las preguntas. Otra cosa es que, bueno, este año queremos seguir fomentando la participación de los estudiantes de posgrado, así que les pedimos a los PIs que estimulen a sus estudiantes a hacer presentaciones. Nos parece que y también para todos enterarnos y para recibir aportes a lo que están haciendo. También vamos a mantener la figura de moderador. La idea es que quien presente a la persona que va a dar la charla también vaya pensando, tenga preparadas algunas preguntas y facilite la discusión. Y bueno, y aprovechamos esta instancia también para algunos avisos, ya que somos bastantes. Este año tenemos la visita de Paola Pino. Paola se puede parar y saludar. Vení acá si te ven a los que están en YouTube. Un segundo. Y Paola viene de Pasteur Networks, habla español, y mañana va a dar una charla a las 9:30 en este mismo salón para explicar un poco el motivo y el contexto de su visita. Va a estar un mes en Uruguay con algunas entradas y salidas, y hablar un poco de lo que está apostando Pasteur Network. Estaría bueno, es una charla corta, media hora, estaría bueno, informal, para enterarse un poco de por qué está acá y qué cosas están pensando. Así que mañana te esperamos. Y bueno, ahora le doy la palabra a Carlos Robelo, que va a presentar a nuestro primer invitado del ciclo.
Bueno.
¿Algo más?
Sí. Muchas gracias para la Comisión Seminarios. Y voy a ser breve, cortito. I want to thank Dr. Timothy Wade that is here. The main reason is because we have a PTR project with the laboratory of Lionel Malacrida, the Timothy Wade laboratory, and our laboratory related with the interaction with what is a mitochondrial process and interaction between Ciclo and human or mouse cells and the mitochondria. Timothy Wade has done his graduate and postgraduate studies in the McGill University in Canada, Montreal. After that, he did a postdoc in the Max Planck Institute in Germany, and now he has his own laboratory group, G5 group, in the Pasteur Institute in Paris, that the name generically is the Mitochondrial group. Thank you very much. He understands Spanish, so the questions are in Spanish, and he will.
Or in French, as you like. The language you want. All right, I hope you don't fall asleep. And I hope for you too.
Anterior.
I think it's good to keep with the diet. Okay?
It's fine, it's fine. I'm sorry for the delay. First of all, I'm going to give the presentation in English. Yesterday I was at the School of Medicine, and I offered the questions could be in Spanish. I understand Spanish. I don't speak very well, but I can understand. So for me, the most important is I want to share with you some of the published and unpublished work we've done that's going to be in the context of this project quite interesting, and maybe interesting for some of you who have a peripheral interest in mitochondria. And so please feel free to interrupt me at any point. Raise your hand, yell out if I can't see you in the dark, and really feel free to ask the questions in however we find a way. The most important is that there be no barrier to asking questions that we can discuss. So for me, I'm very, very happy to come here. It's my first time in Uruguay, first time in Montevideo, and to see Mr. Pastel de Montevideo, and to learn about the science that you're doing here. And it's been cool to see the pictures on the internet, but there's no replacing the human interaction connections we can have here. So I'm very, very happy to be here and to tell you about some of the work that we do. So my lab is the Mitochondrial Biology Group. That was the name we gave to it when we came in 2018 in Pastel, and we work on all aspects of mitochondria. Well, not all aspects, but we're the only team that really works on mitochondria. Uh-oh. Uh-oh. All right, we're in business. So broadly speaking, my team is interested in lots of things that deal with mitochondria. In terms of genetic diseases, where there are patients that suffer from multisystemic disorders where there's mutations in mitochondrial genes. In animal models of disease, we don't really care about which tissue we target, whichever one has an interesting phenotype and interesting biology. But I'm going to tell you about a story about the heart today. And we've gotten interested in microbes and mitochondria, bacteria, SARS-CoV-2, and now T. cruzi. So this is globally what we do. And when we think about mitochondria, we often think that mitochondria in English, at least we say the powerhouse of the cell, right? It's what makes all the energy. And that's what you can see on Twitter. That's what you can read in textbooks. And if you ask my mother, this is what she probably would say, right? And it's thanks to this guy here who says, you know, we've got the central role of ATP generation. And this is quite interesting because Louis Pasteur, even before he knew what mitochondria were, he discovered the importance of mitochondria in metabolism. So he'd done this experiment. This is something they call the Pasteur effect, where he grew yeast in the presence or absence of oxygen. And he saw that the growth rate was different. So in the presence of oxygen, the yeast would grow and ferment and grow at a much greater rate than they would in the absence of oxygen. And this is even before we knew what mitochondria were. So this is the argument I give when they ask me, why are you at the Pasteur Institute? I said, well, he knew about mitochondria long before we knew how they worked. In the context of energy production, we think of mitochondria in the powerhouse of the cell because of the respiratory chain, which is the first 4 enzymes that you see here, multisystemic, multi-protein complexes, and the ATP synthase. And very broadly speaking, this is very complex. Lots of people study it, lots of proteins, lots of regulation. But what you need to remember, or just think about, and a bunch of Nobel Prizes to boot, is that the respiratory chain shuttles electrons eventually to molecular oxygen, and that's what to make water, and that's what consumes the oxygen. And as a consequence, protons are being pumped into the intermembrane space. You have the inner membrane and the outer membrane of mitochondria. And these organelles are, of course, indispensable in every nucleated cell of our body. And with this proton motor force, it's then pummeled through the ATP synthase. The ATP synthase turns as a rotor, and this makes ATP from ADP. And I like to think of it in a much simpler way, which is the respiratory chain makes protons that are then pumped in through complex time. So this really needs to work. And another analogy I give when we talk to people that are not used to looking at this is like a hydroelectric dam. You've got this big reservoir of protons that literally has to turn a rotor, and the ATP synthase is a rotor. Complex type looks like a rotor when you look by cryo-EM electron microscopy at the crystal structure. And that is generating the ATP. And so that you can also appreciate from this analogy that this process can't have any leaks. This process needs to be coupled. And so we look at proton pumping and ATP production is being really coupled within the system. And this is going to be important for the rest of the talk. So we've got an interesting structure of mitochondria, and the machines that make the ATP, the SOx-PHOS system, and the TC, the respiratory chain, live mostly in the invaginations of the inner mitochondrial membrane, something we call cristae. And so the cristae serve to increase the surface area of the inner mitochondrial membrane, which gives real estate for the machinery to be assembled and function. So the more cristae have, the more capacity for energy. That's really important as well. Cristae do other things, and we'll get into that later on in the talk. So this is the structure that you can see of mitochondria. And what's also interesting to know is that there's machinery that makes some of the proteins of this SOx-PHOS system. Now, there's lots of proteins in these complexes, and 13 of them are made and translated by mitochondrial ribosomes inside mitochondria. Transcribed, there's a machinery for transcription, and there's even its own mitochondrial genome. And the genetic code here is a little bit different than the genetic code of the nucleus. So we're not talking about the same amino acids that are made by the same codons. And this looks a lot more like the bacterial system. For those of you interested, you'll find some parallels between mitochondria and bacteria. So we know today of about 1,500 mitochondrial proteins that are needed for its function. And 13 of those, I told you, are encoded by mitochondrial DNA, which means the rest are encoded in the nucleus. They're made inside azelaic ribosomes. They need to be imported into wherever they're supposed to be and function inside mitochondria. And today we know about 400 different genes that are mutated that give rise to these very complicated diseases that we really don't understand. I'm not going to talk about the diseases. We can discuss this if you want later. They're very difficult to diagnose. Some people in Montevideo are doing this with the School of Medicine, but it takes years for the to come. And even with genetics, we're only at about 50% diagnosis with a molecular defect. But you can find mitochondrial dysfunction basically in almost any disease you want to study, right? And we have neurodegeneration, idiopathic forms can have a mitochondrial component or mitochondrial dysfunction, renal failure, cachexia, heart failure, which is our predilection, and a number of different infectious diseases have been associated with mitochondrial dysfunction. Whether they are the trigger for the disease or they contribute on some ways, not so clear, but nevertheless, we can see mitochondrial dysfunction in kind of these more acquired common disorders than the rare ones we study. So if anything, I can leave with you two messages. The first is that mitochondria have a biosynthetic role. We can divide it up into kind of two categories. And this would be involved in the generation of ATP, but they also signal, and this is going to become important later. ATP synthesis is the powerhouse of the cell that I told you about. Metabolism of nucleotides, lipids, and amino acids, and iron sulfur cluster, all really, really important for mitochondria. And signaling of programmed cell death. This is important because there's molecules that are sequestered inside mitochondria that need to stay inside mitochondria. They're liberated. These proteins will moonlight and actually turn on caspase and trigger programmed cell death. Stuff that was discovered in the 1990s. Reactive oxygen species signaling, which of course is very popular in Montevideo as well. And most recently, innate immune signaling, which is something that we've gotten into just kind of by chance. So if you look at mitochondria from an organism, but in different cells, you'll appreciate very rapidly that they look different in different cell types. So you can see in the heart, you have packed cristae, those intermembrane invaginations where the SOx-PHOS machinery lives. They're jam-packed in the heart and really kind of scant in the liver and in the kidney and in other cell types. And so mitochondria look different and have different functions in different cell types. You can do this by transmission electron microscopy quite nicely. But you can also look by fluorescence microscopy, and some of you here are interested in that. You can label mitochondria with fluorescent probes, transgenically, or molecules that will target to mitochondria and be fluorescent. And here you can see mitochondria in green of mouse embryonic fibroblasts that we isolated, and the nuclei is labeled in blue. And you can take cells from the same mouse, cardiomyocytes, they look totally different. So now the mitochondria are labeled in red here with TMRE, and they're jam-packed full of cardiomyocytes. There's basically no space for anything else left in the cardiomyocyte. And that same mouse, if you look at the primary hepatocytes in the liver, they look again quite different. So for you, the take-home message is different cell types, mitochondria will look different, they'll do different things, and we maybe shouldn't necessarily expect them to have the same functions. So when I started the lab, we were interested in mitochondrial morphology. We were struck by the beautiful shape that mitochondria can take. You can see here in human fibroblasts from a healthy child. You have this beautiful reticular network, and there's the nucleus. And upon stressing, you can see that they respond exquisitely to this poison of the respiratory chain, antimycin A. And you're going to start to see here that mitochondria, within a few minutes, they start to fragment. You can see this fragmentation here. And it's so dramatic here that these mitochondria fragment and are actually removed by an autophagic process called mitophagy. There's a lot of correlations between the shape mitochondria have and their fate. The shape mitochondria have is balanced by rates of mitochondrial fusion and mitochondrial fission. And in a normal situation, you have a balance of both. But if you tip to balance it between having more mitochondrial fusion, you have hyper-elongated mitochondria. And if you tip the balance to more mitochondrial fission, you have mitochondrial fragmentation. And again, this has been associated and can be triggered by a number of different stress conditions that might be interesting for you here in the audience. These are kind of pathological stress conditions that we in the lab as best we can. Can be instigated also with genetic manipulations. We can knock out genes and remove proteins that play important roles. But these processes are also important in normal physiology. So for some types of cell differentiation, you must have elongation of mitochondria, without which you don't get the proper differentiation. For mitosis to occur properly, for cells to divide and divide up their organelles, they must therefore also divide up their mitochondria. If they don't do this, the cell growth is slower, mitosis is blocked, and you have spindle checkpoint defects. So there's a physiological for fission and fusion as well. Here are examples where we've knocked out proteins that are essential for mitochondrial fission, and mitochondria become much longer, as you can appreciate here. Or we knocked out a protein that is essential for mitochondrial fusion, and mitochondria continue to divide, and you have mitochondrial fragmentation. So when I started the lab, we were interested in understanding molecularly how this worked, because we knew the proteins involved in fusion in the inner and outer mitochondrial membranes. We knew that DRP1 was required for fission on the outside of mitochondria. We focused our attention at the inner mitochondrial membrane because nobody knew how this worked. No dynamo-like GTPases, no machinery required for membrane dynamics had ever been identified. So when I started, we decided to pick, well, we did some screening approaches, but one candidate that we picked was a protein called MTFP1. Now, on paper, it sounds great because it's called mitochondrial fission process 1. We could localize it to the inner membrane, so fantastic. The grant was funded, we're happy. And this was our inroad to try to understand how mitochondrial fission at the inner mitochondrial membrane could be regulated, because these processes can be decoupled, but they're also highly connected under physiological conditions. So there is some evidence that suggests in literature before we started that modulating the levels of MTFP1 could change the shape of mitochondria. There's some data to suggest that taking away made mitochondria longer and overexpressing it made mitochondria shorter. And this is exactly what you would expect to find if you had a mitochondrial fission factor. This is what we found with DRP1. So this fit perfectly. And so what Armenia did, Armenia Donnarumma, who I say is the second most famous Donnarumma in Paris. And I say this not jokingly, her cousin literally is the goalkeeper of Paris Saint-Germain. But he came after she started in the lab. So she came to the lab and she was going to study this process. And what she could do, she could stably lentivirally overexpress MTFP1 and she could fragment the mitochondrial network. And then these are the control cells. And so when she started doing this, we realized and started in the lab, well, how can we quantify the shape of mitochondria? You can appreciate that they're different, but how are we going to take an unbiased way to quantify this? This was really important when we started the lab. And so when we were faced with this question, you could either segment the mitochondria, measure the length, the width, or their position. But one of the other things that we wanted to approach is to have a more facile and unbiased and rapid way of doing this. So the approach we took and developed and is now published was to do automated spinning disk confocal microscopy in 3 to 4 well plates. So there's automated imaging with a confocal microscope. It finds the Z position, takes all the images, and then we use supervised machine learning to quantify mitochondrial shape. So because we knew the genetic manipulations that could give us different classes of mitochondrial shape, we would then go and teach the machine, this is a cell with hypertubulated mitochondria, this is a cell with tubular mitochondria, and this is a cell with fragmented mitochondria. And then we could quantify the unknowns. And this worked rather well, I would say. So Armenia then took this approach, this was a totally other paper, but she took this approach to see what would happen when she compared the wild type mouse embryonic fibroblasts to the knockouts of MTFP1 that we had made in the lab from a conditional mouse model, which actually now is here in Montevideo, in the institute upstairs somewhere, I hope. So she looked at these mitochondrial morphologies. When I looked at them, I said, there's not so much to write home about. Let's do the supervised machine learning quantification. There was an effect. It was very modest. So if you looked at the hypertubulated form, which is what we would expect in the knockout, there was a statistically significant increase, but honestly, nothing to write home about. Not a huge effect that I would base the future of my lab on. Nevertheless, it was there reproducibly. We could see it in a knockdown. We could see it in a knockout. Other people had seen it. So, okay. So the prediction is if you have a fission protein that's absolutely indispensable for mitochondrial division, you knock it out and you try and induce fragmentation, you shouldn't be able to do that anymore. That's true with DRP1, that's true with many other proteins that are essential for fission. So she did an experiment with a bunch of different triggers that cause fission. And the bottom line is she could induce mitochondrial fragmentation, here shown in red, in all cases. So in the cultured cell systems we had, MTFP1 was not required for mitochondrial division. So then, what does it do? That was the next question we were left with. So those kind of sucks because we thought we were going to start working on mitochondrial division, but then we were taken in another direction, but such is life. So one of the approaches we had, which was not super intelligent, but did lead us in a correct direction, was to start knocking it out in different tissues. And so we generated a conditional mouse model and used a Cre recombinase to specifically knock out this gene at birth in cardiomyocytes. And the idea was that we were going to target tissues with lots of mitochondria so we could do biochemistry. And the heart is a great place to start. So this is the approach. We could knock out the gene nicely, which you can see here by Western blot or by legal-free quantification proteomics by RNA-seq. And then we decided to study what would happen to this mouse. So for the first few months, the mouse was totally fine. There's no phenotype. The morphology of the mitochondria wasn't bigger. These mitochondria from the knockouts weren't longer as you would expect. In fact, if anything, they were a little bit shorter. So even in vivo, we didn't have any evidence for mitochondrial elongation. Again, MTFP1 is not required for fission. So then what Herminia does, she's a cardio background, she did longitudinal echocardiography, which allows you to measure the structure and function of the heart. And she monitored these guys over time. And what you can see here in the echo is in the late stage animal, you can see that the heart isn't beating. This is the view of the left ventricle, which is the part of the heart that's pumping oxygenated blood to the rest of the body. It's not pumping well. And you can actually make really nice quantifications of cardiac function that are declining over time. And structural remodeling of the heart, you can have the thinning of the posterior wall. The muscle is actually degrading and the cardiomyocytes are lost, and that's why the muscle becomes thinner, and you can measure this. And then the left ventricle gets bigger and bigger. It's trying to compensate and becomes more and more dysfunctional. And this is something we call dilated cardiomyopathy. This is number one cause for heart transplantation in France. I don't know in Uruguay with the rates of DCMR. And this gives rise to heart failure. And this was a really nice model. So what we see in our model is very similar to what people see in humans, for which really the only recourse is transplantation. It's a thinning of the muscle walls here in the outside and the inside, and a bigger left ventricle. We saw fibrosis, cardiac cell death, the regulation of oxphos was going down, and mitochondrial respiration was going down. So all these defects are very common in heart failure. So this didn't really tell us much. And you can see that the mitochondria didn't look like they were in great shape in the failing heart. So one of the things that we had seen was that the cells of the heart were dying, or at least that's what we thought, because they were disappearing, and we could even see that the markers of the cells of the heart were floating in the blood of the mice. So we decided that we wanted to look at cell death, but before the animal got sick. And most of our studies focused on the presymptomatic phase. So what Herminia did is she took healthy presymptomatic mice, she did a Langendorf-free system to isolate the cardiomyocytes, and she put it in the high content screener to image the mitochondria. And she could image the mitochondria. And because cardiomyocytes have this very particular shape, and they don't look like most of the cells you work with, they're binucleated and they're full of mitochondria, we could use this to teach the machine what a cardiomyocyte looks like. And when we did this, we could actually see all the cardiomyocytes from the wild type of the knockout and ask what happens when we trigger cell death. Do we see a differential response in cell death sensitivity? We can do this because when the cells die, they lose their TMRE staining, they lose the mitochondrial memory potential, and they lose their shape. And you can see this when we hit them with doxorubicin, which is a cardiotoxic agent. You can see that the knockouts are dropping off over time, much faster than the wild types. And the cool thing here is that we can do this kinetically and come up with cell death curves. It's actually quite dramatic at the end. And this is the quantification of the data. When we hit them with doxo, the knockouts here in red die much faster than the wild controls. Now, these come from mice that have normal cardiac function. Clinically, or in terms of imaging capacity, they're totally normal. They have normal cardiac function. So this doxorubicin sensitivity actually turns out to work in vivo. You can inject them with doxorubicin and they develop heart failure faster than the wild types. The cell death sensitivity seems to be also something that is not specific to doxo, to other cell death triggers. And so we had in this model, in the presymptomatic mice, an increased sensitivity to cell death. So something was going on with MTFP1 and cell death. So I've got to tell you a little bit about mitochondria and cell death. I told you that they signal and that there are molecules inside mitochondria that can be like SMAC Diablo, cytochrome-C and IF that can be released under certain conditions. And this can happen when pores form on the outside of mitochondria. And there are a number of events and molecules and mechanisms that lead to the formation of pores we can get into if you want. When those molecules leave mitochondria, they can turn on caspases and lead to apoptosis. And so membrane integrity turns out to be really, really important in the context of signaling and cell death sensitivity. So we have dyes in our cultured cell system that visualize the activation of caspases 3 and 7. So normally they're not turned on, right? Because there's no cell death. And what we can do is we can take wild type and knockout MEFs. They're not the cardiomyocytes, but the MEFs, which are a little bit easier. Label them with a caspase dye, label them with nuke blue, which labels the nuclei, and then do the same type of imaging. And so when we do this experiment, see if the movie can run. What you'll see here on in the knockouts is it's going to should start lighting up like a Christmas tree. There we go. A little bit faster than you see in the wild type. And you can start to see this kinetically over time. So this experiment gets done, we image every hour for 24 hours, and then we can do in multi-wells the kinetics. And this response we can block with an inhibitor of caspase. So then we know it's caspase dependent cell death. So when we do this type of imaging, we can get cell death curves that look like this. So again, the knockouts die faster than the wild types. This response can be blocked by the pan-caspase inhibitor QED. So it's really caspase dependent cell death, like apoptosis. It's true for actinomycin D and ABT737. It's also true for staurosporin. So these cells are intrinsically dying much faster when you trigger them, but under normal conditions, they grow just fine, they respire just fine, there's no problem. So there's intrinsic sensitivity. So then how do we figure out how this works, right? We have this protein inside mitochondria, MTFP1, not involved in fission, seems to be involved in cell death. What's the way forward? So one approach we took was we generated a mouse model that expresses a flag tag version of MTFP1, so we can pull on it. And we expressed it specifically from the ROSA 26 locus in cardiomyocytes. These mice are normal, they don't have any defect, but you can see a modest overexpression here of the transgenic version of MTFP1. Then we isolate the mitochondria, we do a co-IP, and we define the interactome of the mitochondria that express MTFP1 to ask what are the proteins that interact with MTFP1. And we found proteins that were involved in a process called the mitochondrial permeability transition protein. I'll get to that in just a moment. Suffice it to say that these proteins that we found are involved in this type of complex that we still don't fully understand, whose opening leads to swelling of mitochondrial rupture and release of molecules that drive cell death. So the PTP is something that was described pharmacologically. There are chemical inhibitors of this that work very nicely. Molecularly, we're still kind of not sure exactly what are the key constituents of the four. Nevertheless, this PTP seems to its regulation is well known, and our protein MTFP1 seem to interact with regulators of this. So this is the road we took to try and understand what happened. So MTFP1 not only could physically interact, but it forms a complex with PPIF, which is cyclophilin D, and ANT1, these regulators. And the way we could show this is we can do something called a blue native page. So you can run an SDS page, which denatures your protein. But you can also run acrylamide gels in non-denaturing conditions, so you can keep your complexes assembled. So that's what we did in the first dimension. Armenia did in the first dimension. I didn't do anything here. And then in the second dimension, she ran the SDS page, and she could see a co-migration of ANT1, PPIF, along with MTFP1. So it seemed like there was a complex of MTFP1 of, let's say, 150 kilodaltons being formed, which incorporated proteins that physically interacted with the protein. So to test whether MTFP1 was involved in PTP regulation, we did an assay that's relatively simple, in which you can promote the opening of the PTP using a variety of different stresses. We chose calcium. And this causes mitochondria to swell and change their light scattering. So you can actually measure light scattering on the y-axis. And you can see that when you trigger PTP opening, the wild types in green, or the controls in green, scatter and swell. And this is significantly faster in the knockout mitochondria. Again, these mitochondria come from pre-symptomatic mice. So it's not a consequence of the heart failure that's the difference here, right? This is an intrinsic capacity to respond differently. And this response could be blunted using the drug that prevents PTP opening. So we know we were looking at PTP opening. You can do this for mitochondria from the heart. You can also do this for mitochondria from the MEFs, from the control and knockout MEFs. And we could even genetically ablate PPIF and rescue this phenotype so that we knew it was a PPIF dependent mechanism. So it's told us that somehow PTP is being regulated by MTFP1. And this makes sense because they're all kind of located in the same part of the membrane. When we looked at cell death, we saw that the triggers of PTP opening were enhancing cell death by live cell imaging with H2O2 or doxo in the MEFs. And if we knocked out PPIF, we could rescue this as well. So again, we had a nice link between PTP opening sensitivity, MTFP1, and cyclophilin D dependent opening. So this told us that we had an increased sensitivity to cell death because of MTFP1 that seemed to be regulating this pore. So the model that we had then, and we have now, is one in which we have MTFP1 that seems to be a negative regulator of this pore. When it's absent, there's an increased sensitivity to cell death opening or cell death sensitivity because of PTP sensitivity, because of this molecular regulation that we're still trying to figure out. And this happens in the pre-symptomatic mice. So this is existing really before the animals get sick, and we therefore think this is the trigger or one of the triggers for disease. I started talking in the beginning about mitochondrial respiration, and for a reason, because we saw respiration decline as the heart was failing, in fact, as many organs have failed. And so we wondered what would happen in the pre-symptomatic mice. I told you that I can tell you that the cardiomyocytes contract normally, so we can field stimulate them and they have no contractile dysfunction. Morphology is normal, gene expression is normal. What about respiration? It's a very logical thing to look at. So we wanted to look at mitochondrial respiration, and I remind you once again that the respiration is very, very important. This process of coupled mitochondria, where proton pumping and ATP synthesis are coupled, is extremely important in the heart. So the heart needs and makes 6 kilograms of ATP a day. It's a tremendous amount of ATP. So any sort of leakage in this system theoretically could be catastrophic. Right? And we know this from human studies. And so to revisit this question of proton pumping, one of the things we looked at was mitochondrial function and respiration. And I'm going to just give you the punchline. We could see that the assembly of the respiratory chain, and here we're looking at a 1D blue native, was normal. But in fact, the membrane potential, this coupling was affected in the knockout mice in the pre-symptomatic stage. There was this change in membrane potential that was due to a proton leak. So somehow MTFP1 loss didn't really affect the capacity for the maximal respiration rate to be achieved, but the membrane potential was lost and somehow protons were leaking. And because we've done the proteomic studies, we found that MTFP1 was interacting with a protein called ANT1, and we had a specific inhibitor of that. And the use of that inhibitor was able to rescue that proton leak and that lost membrane potential. So we have this loss of membrane potential and uncoupling of mitochondria because under normal conditions, when MTFP1 is present, most of the protons are going through complex 5 to make ATP. But when it's absent, now all of a sudden some of these protons, for reasons we don't understand yet, are leaking through ANT1 and uncoupling the mitochondria. Now, most tissues don't care, but the heart is really sensitive to this, and this happens before the animals get sick. So right now, work in the lab is trying to figure out, are both the mechanisms that we link to MTFP1 important for the manifestation of cardiomyopathy? Is there one that's more important than the other? Can we dissect these mechanisms in vivo? And this is what we're looking at. So which is with the last few minutes, and this is kind of what, in a way, brought me to meet or to get in touch with Carlos and Leonel and explore this possibility. When we looked at the failing hearts, we did all sorts of omics. We did metabolomics, proteomics, and transcriptomics, honestly, because we didn't know what we were doing and we just shot in the dark. And when you don't know what you're doing, you do omics and you hope something comes and falls from the sky and you can learn something. I have to say that we did learn that there was an interesting, from the transcriptomics, was really robust, is that we saw as the animals developed cardiomyopathy, there was this huge inflammatory response that we could see transcriptionally. And this was really strange because these animals in Paris are housed in a very clean facility. It's not an infectious, as far as we know, not an infectious animal facility. So there's no reason for there to be an infectious output. And so we did all sorts of analysis for RNA-seq, and one of the pathways that came up was Chagas disease. And Chagas disease, as of course we'll know from the work here, is a disease that is multisystemic but has a clear cardiac component in some patients. And it's a cardiomyopathy. It's an infectious cardiomyopathy. So we saw signatures of myocarditis and sort of innate immune signaling response that was really highly upregulated in these hearts. And so we were intrigued by this, and then this is the kind of the genesis of the project that we've started now and kicked off this week here in Montevideo. But we were really intrigued by this response and this upregulation of innate immune signaling. And this is what we saw. And we've been getting interested in the field because, in fact, innate immune signaling can be instigated by mitochondria. So mitochondria contain their own DNA. It's a circular genome, looks a lot like bacterial DNA. And it's, as I told you in the beginning, essential for the synthesis of proteins required for oxidative phosphorylation, for ATP synthesis. But under some conditions, it can leave the mitochondria and find itself in the cytosol. And because it looks like foreign DNA, like bacterial DNA or other foreign DNAs, it can be recognized by the same type of sensors that are there and have evolved to recognize pathogenic and foreign DNA, such as CGAS, inflammasome can recognize it, toll-like receptors can recognize it, the cell type. This is the common pathway. And this leads to the modification of the STING and the translocation of transcription factors to the nucleus that turn on interferon-stimulated genes and make interferons and the signals through interferon receptor. So we've seen this response in cardiomyopathy, and this happened clearly, this innate immune signaling response. We could also see by increase in cardiac resident macrophages in the heart and cytokine production. So there was something that was going on here, and we wanted to test this. And so what we ended up doing is we additionally knocked out either STING or IFNAR1 in our heart failure mouse model to see what would happen. And what we could see is that when we looked at the knockout that has a cardiac function that's reduced here, you can see it in mode echo, but you can maybe look at the graph. By knocking out either STING or IFNAR, we could increase cardiac function. In fact, the IFNAR knockouts even rescue the lifespan. We were looking at the lifespan of these mice, and we can actually see that increase in cardiac function, this correlates with an increase in the lifespan of the double knockout mice. Here we're looking at the survival curve of the females. So what's actually quite fascinating is those two primary mitochondrial defects I told you about at the beginning in the knockout, the uncoupling of the mitochondria, right, the cell death and the cell death response by the PGP, they're still there in those double knockout mice. So we haven't fixed any of the initial mitochondrial problems, the primary mitochondrial defect that the MTFP1 deletion causes. We've just fixed it pretty cool. But we've fixed the downstream inflammatory problem as far as we can tell. And that seems to be largely sufficient to extend the lifespan and cardiac health, cardiac function of these mice. Now, I should say that the knockout of STING and IFNAR in this scheme is a whole body knockout. So we haven't yet figured out if we only knocked out STING in cardiomyocytes, right? Because our MTFP1 knockout is only cardiomyocytes. Does that rescue the function? So then we generated the STING flox, MTFP1 flox double knockout mice that are specifically cardiomyocytes and also CGAS. And well, next time I come, maybe I'll tell you about that. Okay. So this leads us to like, well, what's next? So our idea is that mitochondrial DNA is being released, right? But how do we look at this? And so I just want to show you a little bit of preliminary data how we tackle this question in mouse embryonic fibroblasts. So what we've generated is a system now where we can express a reporter, it's shown in green, on the outside of the mitochondrial membrane, something we use as OMP25. And we actually have a protein that binds mitochondrial DNA called TFAM, which is an essential protein. And we can co-express these with lentiviral delivery in cell lines. And we can see that, in fact, I don't know if you can see this, is that you have what we have, these little punctate dots, what we call nucleoids in magenta, that are dotting like a Christmas tree the mitochondrial network. What we actually developed with lattice SIM, structural elimination microscopy, with the platform, is a way to visualize the release of mitochondrial DNA. We can stimulate this release chemically, and then we can see that mitochondrial DNA is now no longer retained within the network. So that's really cool. So then how do we quantify this? Fortunately, we have great colleagues that we work with. They're doing some really nice preliminary work, where they've developed, and Stefan has done this in Jean-Yu's team, a Gaussian spot detector to actually detect spots of mitochondrial DNA as in the stimulated, non-stimulated conditions. And using the raw data, they can look at the mitochondrial DNA spot or the mitochondrial network and do an intensity profile measurement and see in the stimulated condition that this guy is not getting the same intensity profile. This can be done also in a transformed, what we call Otsu transformation. And you can clearly see when mitochondrial DNA is outside versus when it's inside. And basically the way they do this, or they're currently working on, is to define a radius and look in all sorts of directions and define a threshold after which we don't detect, let's say, the green mitochondrial network in all sorts of directions. And that indicates, for example, here, that it's outside. So that's what we're working on. And maybe next time I come and speak with you, I'll have a little bit more. And this is what we're trying to now hopefully apply to cardiomyocytes. So if I can leave you with the time I've got, with three messages about mitochondria, which I think is more expansive and more applicable to other cell types and other models that you may be studying, is that mitochondria indeed have multiple functions. And what our job is, or what we struggle with, is trying to figure out which of those functions is really important in which cell type and when. And it's not necessarily a binary response, but it's something that we think about more globally when we think about mitochondria. Individual proteins can have multiple functions within the organelle. MTFP1 is a great example, but I think it's true for most proteins. MTFP1 regulates cell death by PTP opening, and it regulates bioenergetics. It seems to be in a cell type dependent fashion. And finally, what I showed you is mitochondrial dysfunction can trigger a downstream maladaptive response. And that may turn out in some situations, and even in humans, to be even more important than the primary mitochondrial trigger that was caused by the genetic lesion. with that, I'd like to thank certainly Armenia, who did the bulk of the work and was assisted by Elodie, who's our lab manager and technician. Wonderful members in the lab, our collaborators in Würzburg, who did experiments that I didn't get a chance to show you today. Fantastic core facilities that we have in Paris, really fantastic, without which we wouldn't be able to do 90% of the work that we're fortunate to be able to show you today. Great funding, great food, great colleagues, and thank you very much for your attention.
Thank you very much.
En castellano, en inglés, o en francés, en alemán, como quieras.
Hi, great talk, thanks. I have a question regarding the rescue of the phenotype of the mice. So you know that the mice still have the bioenergetic problem in the mitochondria, right? So it's more like a signaling than bioenergetics, the triggering of the function. So do you know how the heart or the cardiomyocytes are coping with this uncoupled mitochondria? What is, if there is any metabolic shift in the cardiomyocytes that's helping them to keep running?
So cardiomyocytes are quite unique because most of the energy comes from the metabolism of fatty acids, right? About 70%. And so the bioenergetic defect still persists in these double knockout, let's say, rescue mice. It's not perfect, they still die. And our feeling is that this is because the primary mitochondrial defect still somehow contributes, because the mice die of pulmonary edema. So basically the lungs fill up with fluid, and this is because of the cardiac dysfunction. This is clear, you've shown this. So my feeling is that there's for sure a metabolic switch that we see by RNA sequencing. So we can see a fetal gene expression program that's consistent with more glucose oxidation and more metabolism you would see in a fetal heart, and not the one in a normal heart. And this is also consistent with heart failure. When heart failure manifests, we see this. So I don't exactly know if they can compensate, but what we're looking at now is we've seen that or Armenia's seen that the phenotype in the males and female single knockout mice is not the same. So the males are more sensitive to this knockout than the females, and that's also true for the double knockouts. And if you look at the female and male wild type hearts, you actually see that the OXPHOS proteins aren't expressed in the same way. Their substrate use is different, the metabolomes are different, right? Heme is a #1 dysregulated, differentially regulated metabolite, and heme is absolutely essential for complexes of the respiratory chain to function as a prosthetic. So there's got to be some sort of compensatory reaction and some sort, not compensatory reaction, but there's some reserve of mitochondrial function, past which the mice then suffer. That's why it takes them 18 weeks to get to heart failure. And what I talked about yesterday, we did the same knockout in the liver. The mice live fine. Liver, in fact, they do better if you give them a high-fat diet. So this uncoupling seems to be really specific to the cardiomyocytes. We could not find this in cardiomyocyte cell lines, nor in mast, nor in HeLa cells, nor in HeLa cells, or any other cell type, HUH cells. None of the cells we looked at do we see uncoupling, only in the primary adult cardiomyocytes. So I think it is something very specific about this cell type, which makes it sensitive to uncoupling.
So, I was wondering, when you try isolating cardiomyocytes from the double knockouts and see whether in culture they behave the same, because right now there's a paper from Spain. They show that the cardiomyocytes can pack positive mitochondria and send it to macrophages. And I was wondering whether maybe DNA leakage can happen in the macrophage.
So 2 years ago, Andres Hidalgo from CINI came to talk about his macrophage story, where the macrophage, so mitochondria exit the cardiomyocytes and the cardiac resident macrophages eat them up, and that is a really important process for cardiac function. And so I asked him about this because we shared some of the data with him when he came. And I asked him, could we test this maybe in vivo even? Could we find a way to get rid of cardiac resident macrophages specifically and see what the outcome would be? Is this something that's mediated by the cardiac resident macrophages? So in a grant that was never funded, I wanted to do this culture experiment where we take co-culture cardiomyocytes from mice and macrophages, maybe bone-derived macrophages, and ask the question whether they could talk to each other. Because we still don't know, we have this nice rescue for the double knockouts, but we still don't know if this is a cell autonomous or non-autonomous process. So we don't know if this inflammation is intrinsic to the cardiomyocyte, which could be, we see this in other cell types, or whether it needs the surrounding tissue. And so your suggestion is a very good one. We haven't yet been in a position to isolate the double knockout cardiomyocytes. We're still hoping that the mice will live, and if we isolate them, of course, we reduce that likelihood. But I think it's a really important experiment to do to see really how different those double knockouts are. And I'm hoping that our double knockouts specific to the cardiomyocytes, because I mentioned that the other knockouts of the inflammatory process is a whole body knockout, will give us a little bit closer to answering your question.
Nestor, I just wonder whether you have investigated any link between ferroptosis, STING1, and I mean the change of events you are seeing, you know, by consequence of this mitochondrial dysfunction.
So we are very intrigued by ferroptosis, very intrigued with ferroptosis, and we have a project on it at the moment. So we have, I don't know, we're looking at it this week. So I don't know what the answer what the answer will be. When we look at mitochondrial defects that drive ferroptosis in the heart, the type of cardiomyopathy is much more severe. And so Thomas Langer had this beautiful study where he looked at HOMA1, W1, and the mechanisms for how mitochondria regulate ferroptosis. And then the COX-10 knockout mice, and so COX-10 is a protein required for cytochrome C, whose absence triggers ferroptosis in the heart. That cardiomyopathy happens after 2 or 3 weeks. So it's very severe, and it's a different type of disease. We have a much more chronic, kind of progressive, relaxed, I can say, type of cardiomyopathy, where we haven't yet excluded ferroptosis as a mechanism for death, but we have no evidence to suggest that it's involved. And it would be great to do this. Now, the struggle is there's an inhibitor for ferroptosis, right? So you can use ferrostatin to block this approach, to block this process. I don't know if it's amenable to an in vivo injection experiment, but this is now something we're doing to test all the different types of cell death in our knockout models, but first in cultured cells, and see if they translate them to the cardiomyocytes and eventually in vivo. But ferroptosis is also involved with changes in ROS and these types of things. We explored ROS early on. We did in vivo measurements of ROS with MitoP and MitoB, which is a mass spec kind of probe that allows you to measure ROS. We didn't see any differences. We saw no differences in ROS production from the mitochondria. And when we treated the pre-symptomatic mice with MitoQ, which is an antioxidant directed to mitochondria, we didn't rescue the phenotype. So we don't think that ROS is really involved here, at least in our model. But it'll be interesting to see what happens with the ferroptosis. I agree.
Thank you. Wonderful talk. I was wondering from the clinical point of view, you said there is a lot of people in France who have cardiomyopathy.
Yeah.
Is there any, do you know if there is any link between this disease of this problem and mutations in this gene?
Right. So we looked hard. We never found patients that have mutations in this gene that suffer from any type of disease, not for lack of trying. This gene is located on a chromosome that's subject to microdeletions sometimes. So then it's really hard to figure out if it's this gene or not. But more globally, we had a project with some researchers in Spain and in Italy, where we asked the question, could we look at all types of idiopathic cardiomyopathies that have been ever sequenced? So there's certain genes that are involved in this, and they come out quite quickly. There's about 400 patients that had never been diagnosed, but had the exomes. And Sylvia looked for SNPs or for, let's say, pathogenic mutations or possibly pathogenic mutations in mitochondrial gene list. And we never found anything really convincing. And I'm not talking just about MTXP1, but any gene that we thought was involved in mitochondrial, let's say, quality control for mitochondria. So that was a few years ago. So we have no evidence that in idiopathic DCM or this type of disease, we can pinpoint specifically a mitochondrial protein. That doesn't mean that you don't have cases of mutations in mitochondrial genes that have, among other phenotypes, DCM, but not the kind of DCM that hopefully you won't ever face, but this type of more specific type of disorder that seems to be more common in the population.
Thank you.
So yeah,
Thank you very much. You mentioned that you saw a sex difference between your mutants, the effect of the mutation, right? Did you look any further to explain was it a survival?
Everything.
Eh?
It was everything, survival, cardiac function. This is something that Armenia is proposed as a project to try and get a permanent position, which I think is fascinating because we have really nice evidence that in fact the bioenergetics are different between wild type males and females. That could fully explain why uncoupling of mitochondria would show a sex difference. The notion that there is a sex difference between male and female mice, even in humans, although it's more complicated because we have the context of gender, but at least in the mice that we have in the lab, is not new. Like you see this in adipose tissue, you see it in many different liver disease, so it's not a novel concept, but it's something that is testable, right? You can do an ovarectomy in a female, you can inject estrogen in a man, you can see how this responds, and so I think that that's the way to go where we can get at the molecular mechanism. And so estrogen regulates proteins that are essential for mitochondrial biogenesis. There's an estrogen response elements in nuclear encoded genes, and even the estrogen receptor has been proposed to target 2 mitochondria. So there's lots of kind of circumstantial evidence to suggest that we should explore this. And in the beginning, I mean, I told Armenia, forget the female. The females are complicated. Just do the males. Everybody's doing the males, right? And if they're hormones, it's going to be complicated. And she was right not to listen to me. She was really right not to listen to me because she wouldn't have found that we wouldn't have. I mean, I didn't do anything. She wouldn't have made this observation. And if you look in the literature, even in our fields, that in general, most studies are in male mice, right? Unless you're looking at a female reproduction, it doesn't really make sense to look at male mice. Although I don't know if you read the paper yesterday in Nature where they can take DNA cells from a male mouse and turn it into ovocytes and oocytes and then turn it into a real life animal. But the problem is that most of our research is focused on male mice, and we're missing half the picture. So hopefully, Armenia's project will get funded and we train back with an answer to that.
Hi, thanks for the nice talk. I was wondering how these genes conserve along the tree of life. What about
So when we looked at this particular gene MTFP1, we didn't know much about it. There's no kind of catalytic domain, and one of the first things that I did was to look at when did it appear. So it's specifically in metazoans only, and you can use gene climb, which allows you to see what's the coevolution with other genes, right? And maybe this gives us an insight into the function of this protein. And so what I was excited about when I started the project, and maybe I wrote this in the grant, was that it co-evolved with SNRS, which is involved in membrane biology. So I thought, oh, fantastic. Then it's involved in fission and fusion because it's a protein, right? So, and the name was MTFP1, so it's fantastic, great. We can sell the project, right? But when we look at the evolution, we don't see, we have no coevolution insights into really the function of this protein. It's really just by doing kind of the bucket biochemistry that we figure this out. And we haven't figured out more function about this protein by looking at different tissues. In the liver, I told a completely opposite story. It's actually super beneficial to knock out this gene in the liver, right? Specifically. And so the only other line about it that I can tell you is we knocked it out in the worm, and the respiration goes up, and the worm lives longer, and we're trying to figure out why the worm lives longer with our collaborators in India. So there is conservation, but it's not really helped us to figure out more than what I showed you today. This is for the heart.
I'll link with that question. First of all, thank you for that talk. I'm wondering, in the term of biogenesis of the mitochondria, when you start about the division of the cell, so you talk about the fusion and fission. So which process is the first one? What is the tendency? Go to fission or go to fusion at the beginning?
That's a really nice philosophical question. I wish I had it. It's kind of like saying, when does life start, right?
Yeah. The question is because in human, you can see several mitochondria, so tend to fission. But for example, in kinetoplasty, what you see just one.
Okay.
The little arch. So it's like the fission is absent.
Okay. So let's start with an oocyte. At fertilization, you have an egg, let's say, depending upon which species, somewhere with 200,000 mitochondria. They're all kind of individual. There's no biogenesis. There's no transcription of any nuclear genes. It's silent. There's maternal factors. There's all this going on. And then you have cleavage of the embryo. And you have just the division every cleavage of the number of mitochondria per cell, because everything's silent, right? And you have these individual mitochondria until post-implantation. And post-implantation, for reasons that nobody's really shown, mitochondrial biogenesis starts again. Because you're getting down to the number of mitochondria per cell, and notably in germ cells, which is important for the continuation of life, where if you go any lower, you're not going to be able to make more mitochondria. You can't make them de novo, right? And so they're still individualized because you need to have them segregated to daughter cells. And then for whatever reason, when you start looking at tissues post-implantation, then you see changes in mitochondrial shape. Then the mitochondria get longer in, let's say, prenatal, let's say the cardiac precursors, right? And not only. So from that perspective, I think that we need them in a rapidly dividing cells to be fragmented. But you can go back and you can look at, depending on the cell cycle, the shape of the mitochondria is totally different. So G1, G0, it's going to be different, and it's going to be, and that's, I mean, it's really dramatic, and I can show you some of those images later. So I think it's a constant process, and I often ask myself, well, then who cares? Is the shape of mitochondria even important, right? So maybe that's a discussion for another time, but I think that when we think about fusion and fission, it's a dynamic process, and the work we've done is look at it at a whole cell level, but I think now we need to look a little bit more deeply and say, are these dynamics the same in all parts of the cell, or are they different depending upon when you look and when and how you look? So I don't have a good answer to your question. All I can say is that fertilization, we should have fragmented mitochondria. If one messes up with the shape of mitochondria, then we have embryonic defects. So it seems to be important at that level.
Just one question. If you want to start with inherited myopathy,
Yeah.
The genes the detected genes are enriched in mitochondria, genes whose main function is in mitochondria.
No, not a little bit, but I wouldn't say specifically enriched. If you have 10% of mitochondrial, not that I can think of. I think the ones that come out to be the most deleterious are ones involved in actual biology of the cardiomyocyte, structural molecules, heavy myosin, heavy, contractile molecules. But I mean, the GWAS studies means that you need to have survived embryogenesis and been born. And so if you have really severe defects in mitochondria affect your heart, you're dead before you're born. So you're not going to have a very mild adult onset disease. But I wouldn't say that there's a striking accumulation of or bias towards these mitochondrial genes.
Okay. Thank you
very much.